Hello, hello, good evening. My voice is audible. Please let me know. Hello, my voice is audible. Please share this link in genetics group. Please, my telegram is right now not working. Please share this link. Please share this link in group. I am unable to, share it in group. If anyone is here, please. सर हेलो सर, मैंने अभी आपको एक लिंक भेजा है, वो ना टेलीग्राम में जेनेटिक्स वाले में डालिए, मेरे से जा नहीं रहा ग्रुप में ना, पता नहीं क्यों, पता नहीं लिंक तो साइन टिक नहीं बज रही। तो मैंने आपको भेजा है, जो भी लिंक भेजा है वो आप ग्रुप में डाल दीजिए प्लीज। जेनेटिक्स वाले में। हाँ, ओके ओके, थैंक यू। सो प्लीज लेट मी नो माई वॉइस इज ऑडिबल और नॉट। माई वॉइस इज ऑडिबल और नॉट। जस्ट शेयर दिस लिंक if possible in the group. Hello, my voice is audible. Any one of you, please reply. Hello. Hello, please let me know my voice is audible, please. Hello. आपने डाल दिया सर? हाँ, कनेक्टिंग कनेक्टिंग आ रहा है, टिक नहीं बज रही। अभी मेरे पास 3 बच्चे हैं जो जिन्होंने वो ही हैं, बाकी आ नहीं रहे। वो कनेक्ट ही नहीं हो रहा मेरा। भेजा नहीं ना जा रहा, भेज रही थी पहले, ये लिंक भी भेजा था आपको, हेलो भी भेजी थी, भेजा नहीं जा रहा। चलो मैं स्विच ऑफ करके भेजती हूँ दोबारा से। Hello, hello. Any one of you, if you are able to listen to me, please send the link in the group if possible. If you are able to listen to me, please send the group in. Please send this link in genetics group because right now my Telegram is not working. So please send the link in group if possible. Hello. Please send the. Yes, the Telegram is not working right now. So please send it in a group if possible. Yes, yes, Telegram is not sporting. That is why I'm waiting here for all of you up to by 6 p.m. But I am not able to get it. Okay, so you are able to hear me. Yes, please, thank you. So it's my request to all of you, if possible, kindly share the link with your friends or either in Telegram if possible, because I am not able to send the link. I am just trying for the last 20 minutes. So please send the link in the group if possible. That is why I always always tell my student to subscribe to my channel so that they will get notification when I get online. So if possible, please share it with all another students so that we will be able to start our session as soon as possible. Yeah, and we have no another how to connect with another students. We have no another link, na? No issues. Then I will start the session. They will just go with the recording, or they will. I will share the link with all of them because it's already 6:17 now. We will wait for 2-3 minutes more. Otherwise, I will start the session. So I'm just waiting for 1 or 2 minutes more. Otherwise, I will start the session. So Prityasha, good evening. If possible, please share the link in the group also. Prityasha, if possible, please share the link. Yeah, we will make it later, but right now, please share this link in Telegram so that students will join. Prityasha, Bhavni, please, if possible, please share this link with your friends. Okay. Yeah, Telegram is not supporting it. I am also trying it from the last 20 minutes, but no. So shall I start my session now? Yes, yes. Please, really, please share with all of your friends so that they will join and I will have to start my session because I'm just waiting from last 25 minutes and I just want to do 2 hours long session today. But I don't know why it always happens that when I am planning something, then something another thing will happen. No issues, but okay. So John, if you have someone along with you, your friends or someone, please share the link with all of them. Please, if possible, share the link. Otherwise, I will start the session. Yes, all of you, please, if possible, kindly share the link with your friends because we are already very late. I don't know why it always happens. So yes, today we have to start with the interactions of a gene, and I have to complete it today, but I don't think so. Yes, so I decided that we have to cover those things, types of gene action, then quantitative traits, qualitative traits, and multiple alleles. I am just trying to do these first. Then we have to cover all these things in detail. So I am just going to start it. So these things we have already done yesterday. The interaction of the genes, what is non-allelic interactions, what are allelic interactions, and what are the non-allelic gene interactions. So I am not going to discuss it again. Yes, so I have to start with these today. This is the normal dihybrided ratio, which is 9:3:1, and today we have to discuss epistasis, complementary genes, duplicated genes, polymeric genes, and inhibitory genes. So what happens in epistasis? One gene hides the effect of other. We will discuss it in detail also. So in epistasis, one gene, it hides the effect of other. So we have two types of epistasis right now. We have recessive epistasis and we have dominant epistasis. The ratio of recessive epistasis is 9:3:4. So instead of 9:3:1, the ratio becomes 9:3:4. And in case of dominant, the ratio becomes 12:3:1. Similarly, in case of complementary genes, the ratio changes into 9:7. Instead of 9:3:1, the ratio gets changes into 9:7. And for duplicate genes, this ratio gets changes into 15:1. So we have to discuss all these interactions in detail. So I have to start with epistasis first. So what is epistasis? So we have additive gene effect. We have recessive epistasis in which the ratio is 9:3:4. And the one more thing is that you have to remember the examples of each gene interaction. So like in case of recessive epistasis, the example is coat color in mouse. So the coat color in mouse is the example in case of recessive epistasis. Similarly for dominant epistasis in which the ratio is 12:3:1, the example is fruit color squash. In case of complementary genes, where the ratio is 9:7, we have to remember the example of flower color in Lathyrus. Getting my point? In case of duplicate genes, we have to remember the example of fruit shaped capsula. So, in case of duplicate genes, the example is fruit shaped caspilla, where the ratio is 15:1. In case of polymeric gene interaction, the example is fruit shaped squash and its ratio is 9:6:1. In case of inhibitory gene action, the ratio is 13:3. So these are the interactions which we have to study. We have 1, 2, 3, 4, 5, 6, 7. So we have to study 7 interactions in which the ratio is not the Mendelian ratio, rather the ratio gets changed from 9:3:1, the ratio gets changes or there are modification in the ratios. Getting my point? So we will discuss all these in detail. In the meanwhile, all of you please try to share the link in the Telegram also, because I am now just delivering the lecture. So I request all of you to share the link in the Telegram also. So these are the gene interactions which we have to study. We have to study all these 7 gene interactions. So the very first gene interaction is additive gene effect. So what is additive gene effect and what basically it implicates? So we have the example of comb shaped chicken. So we have the example of comb shaped chicken. Now what happens in this additive gene effect? Two gene pairs affecting the same character. So basically what happened here? Two gene pairs affecting the same character. Now let me explain you with example. So my voice is audible. So we have gene R, which is rose comb, which is known as RP, capital R small p, and we have another gene which is gene P, which gives us the result of P comb. So what will happen? The dominant alleles of each of these two genes produce separate forms. So when capital R is there, we get rose comb. Okay, when capital P is there, we get P comb. Okay, but what happened? But when capital R and capital P join together, capital R, which is rose comb, and capital P, which is P comb, when R and P join together, they form a new shape which is known as walnut. Getting my point? So both alleles R and P, when they are present together, they give us another character. Instead of rose comb or P comb, we get the walnut comb. So so where R and P are present, we will get the walnut. So where capital R and capital P is present, instead of rose and P comb, we will get the walnut. Okay, so instead of R and P, rose or P comb, we will get the, you can say, walnut. Then another character is there. So when capital R and capital P is present, it leads to walnut. The ratio is 9. When only capital R is present, when only capital R is present, it will give us rose. When only capital P is present, it will give us P. And when only small r and small p present, it will lead to single, or you can say the color will be different from capital R and capital P together. So in this ratio, of course the F2 generation is 9 is to 3 is to 1, but what happens there? There is not only single allele which is dominant, rather both the two alleles, capital R and capital P, they together are dominant. So the ratio is 9 is to 3 is to 1, just like normal dihybrid ratio for two traits. But it is for single trait, which is the comb shape, having 4 different forms. So for a single character, we have two combined together. That means capital R and capital P, when they are together, they give us different color instead of the dominant one. So they are dominant, or you can say co-dominant also. So this is the additive gene effect. When both are present, they give us another character. So this is capital R. When capital R is present, the shape is rose comb. When capital P is present, the shape is P comb. But when capital R, small r and small p is there, the shape is single comb. But when capital R and capital P are present together, the shape is walnut comb. So we will able to get 4 different comb morphologies. Getting my point? We will be able to get four different comb morphologies. So inheritance of comb morphology in chicken was first described by Batson and Puneet in 1906. So this is the very first example of gene interaction. Getting my point? So this is the very first example of gene interaction when two dominant characters, so we will able to get the new character from it. So is it clear to all of you? I hope it will be. If you have any query or question, please let me know. If you have any query or question, please let me know. Let me check it. So this is clear to you till now. The very first interaction is clear to you up to nine up to this. This is a very simple gene interaction. Is it clear to you? So my voice is audible. I hope my voice is audible. So my voice is audible. Please let me know.